Hello friends, welcome back to my channel Xgene and proteinX. In this tutorial video I'll discuss about the beta-galactosidase and also something about white and blue screening. So first you have to know that what is the beta-galactosidase. So beta-galactosidase is an enzyme that hydrolyzes beta-galactosidic bond and forms monosaccharides. So here you see, and also this enzyme also known as the lactase, because it breaks down the lactose, that's what. And if you see here, the lactose is the chemical structure of the lactose is the galactose and glucose, and it joined together by a covalent bond, that is the beta-galactosidic bond. And beta-galactosidase is basically it break down this beta-galactosidic bond and it forms these two monosaccharides of the galactose and glucose. So if you look at the lac operant system, so when the lactose is present in the environment and the bacteria use this lactose for the energy source, and this lac operant it turn on, and the lacZ, lacY and lacA, these genes are gonna start their transcription, and these three genes they encode three different products, and the lacZ, mainly the lacZ, it produce beta-galactosidase or lactase, this enzyme. And the main function of this beta-galactosidase is to break down the lactose into galactose and glucose. Alright. So now I will show you the crystal structure of the beta-galactosidase. So here you see the beta-galactosidase crystal structure, and it is usually the combination of the four identical monomer. It consists of the four identical monomer. So monomer A, monomer B, monomer C and D. So this is 1, this is 1, this is 2, this is 3 and this is 4. And also it contains 5 different domains, and these different, you see so this, if you consider, this is one monomer, and one monomer contain around 5, 1, 2, 3, 4, 5. So around 5 domains are there. And another also important characteristics of this beta-galactosidase is that each tetramer it catalyzes around 38,500 plus minus 900 reaction per minute. So beta-galactosidase it catalyzes these numbers of the reactions per minute. And also another important thing is that each monomer of this beta-galactosidase it consists of around 1 or 2, 3 amino acid. Alright, each monomer it consists of 1 or 2, 3 amino acid. Alright. So I'm gonna move on to the next part. So this is actually the gene cloning part. So here the available vector that these vectors are used for the gene cloning, and in this vector it has a multiple cloning site, and this multiple cloning site contains some restriction enzyme site, and this site mainly used for our gene of interest insertion. Alright, and in this part also it presents the lac operant and also the lacZ and lacZ. So lacZ is basically present near the NCS, multiple cloning site, where we cut our the site and we insert our gene of interest. So when we insert our gene of interest, so this red color part is the gene of interest, and if you see here, when we insert our gene of interest, so there is no lacZ at all, or maybe they are part of the lacZ. So if it is like the full part of the lacZ, this is the lacZ, then when we insert our gene of interest, so maybe some part of the lacZ is gonna replaced with the gene of interest. So then what is the outcome? Then lacZ is not gonna be function properly. It cannot, because the part of the gene is removed. That's what. So now I will show the the white and blue and white screening. So blue and white screening, this is one of the screening process to check whether the gene of interest is successfully transformed in the E. coli system or not. And in this screening system, the Xgene is used. So Xgene is added during the culture of the bacteria, and Xgene is normally the analog of the lactose, but there is some difference between the Xgene and lactose, because Xgene when break down and when break down by the beta-galactosidase, and it produces blue color product. So this is the main important difference in between the Xgene and lactose. So now I will talk about something, this screening system. So this is our recombinant gene. So you see here this recombinant gene already inserted. So this is actually the recombinant plasmid, right? And this plasmid content, as I mentioned in my earlier that slide, that when we insert our gene of interest, so because of that, the lacZ of the gene is partly removed. So that's what the lacZ, this gene no longer works, alright, and no longer produce the beta-galactosidase, because beta-galactosidase is encoded by the lacZ, this gene. So when actually in the transformation system there are the plasmid. So these are the you see, this is the recombinant plasmid. Alright, these are the recombinant plasmid, and also it contains bacteria. So lot of E. coli bacteria, right? These are the bacteria. These are the E. coli bacteria. So you know very well that in the transformation system, not all of the bacteria they taken up the recombinant plasmid. Only the part of the bacteria they taken up this recombinant plasmid. All right. So here you see what is happening here. So the E. coli that taken up the recombinant gene. Recombinant gene means that contain the recombinant gene of interest and the plasmid also. So that content is that kind of E. coli is a lack of lacZ. So this kind of E. coli, they cannot produce a beta-galactosidase. So that's what is leading to this Xgene cannot break down. Alright, so that's what they produce only white color clones, because Xgene is not break down. If Xgene is break down, in that case blue color product is formed, otherwise not. And another part of the E. coli that not taken up the recombinant gene. So that means they have there the lacZ that works properly. That's what they encode this kind of lacZ, this gene, they encode beta-galactosidase, and beta-galactosidase also is translated this product. So whether this beta-galactosidase is present, so upon presence of this beta-galactosidase, the breakdown of the Xgene occurs. So when the breakdown of this Xgene, and it formed blue color clones. Alright, so this blue color clones formed in this when the bacteria in that not taken up this recombinant gene. So here you see in the picture there are two different color of this bacteria, the clones, the blue color and the white color as well. So the white color that I mentioned here, the white color means it indicating the it contain our gene of interest, but the blue color it does not contain our gene of interest. That's what the color is blue. Alright, so this is the main theory behind of this blue and white screening. So now I also want to mention some the application of this beta-galactosidase. So mainly the beta-galactosidase is hugely used in the food industry, and the main function of this is the removal of the lactose from the milk products for lactose intolerant people, the people who cannot tolerate the lactose. That's what the lactose is removed from the milk product by using the beta-galactosidase. And also the production